Welcome you all students. This is the second video. I will be discussing the second aspect of omics, that is proteomics. So in proteomics, we will be studying what is a proteome. So the entire complement of proteins that is or can be expressed by a cell or tissue and organism is referred to as a proteome. And we know very clearly, and this proteomics involves the large study of proteins, their structure, physiological role or functions of an organism under specific defined set of condition in its present state. Is that clear? And the word proteome is a combination of protein and genome and was coined by Mark Wilkins in 1994. Proteomics relies on three basic technologies which are the cornerstone, that include a method to fractionate, that is very, very important. We have to collect the complex protein or peptide mixture. Then we have to spect it, that is called as through mass spec, to acquire the data necessary to identify individual proteins, and next use the bioinformatics tool to analyze and assemble these mass spec data and finalize the structure of the protein. So this proteomic follows these strategies, or you can say steps. First is get the sample, run it on SDS page, so which we'll be studying next year anyway. So either it is two-dimensional page or one-dimensional page, but we do protein electrophoresis, and then we digest this protein into peptides, and we sequence these peptides by when we sequence these peptides, we do the data searching to analyze the protein identification, which is already either submitted in NCBI or protein PDB database, which we'll be studying in bioinformatics. So then we do the degradation of this peptides, that is breaking these peptides by using a methodology called as Edman sequencing. So through this we will build the codons for each peptide, and then we will again analyze these peptides by doing electroblotting. Is that clear? So that is the reverse. So it can be done in two forms. One is either through electroblotting or through digestion by electrophoresis. Is it clear? So this is how the proteomics gets initiated. So to detail it, protein identification. So protein identification, proteins from a polyacrylamide gel by mixed sequencing or MS is depicted. Then we will hybridize that by using CNB-SAC tool, and the resulting peptides are sequenced simultaneously by Edman degradation, as I discussed. Then the MS proteins and angel digested with proteases, and resulting peptides are mass fingerprinted or sequenced. Information of all these methods is used to search nucleotide and nucleotides in protein databases for protein identification. So next year when we do bioinformatics as a project, we will learn how exactly it works. So here you just learn this steps, that's it. You don't have to write this, you have to just learn the steps, what and all is involved. First thing is protein digestion by using electrophoresis, that is through SDS page. Then degradation of these lanes, or you can say fragments, what we have got in SDS page. They will be cut down and they will be degraded using Edman's degradation methodology, and once this is degraded, then we will sequence the codons. Once the codons are sequenced, we will convert the codons into nucleotides, and nucleotides will be will be blasted in the NCBI database to search for protein identification. So this is the methodology which is involved in proteomics. So types of proteomics include protein expression proteomics. The quantity of, here we will study the quantity of the protein expressed, its variable forms, and how exactly the different proteins are present and how they can be compared with each other with respect to the same population or different population or different species. This is called as protein expression proteomics, because the same genes either will be in homologous form or orthologous form. Is that clear? So the type of proteins produced from these genes will be structurally and functionally different. Okay. Next is structural proteomics. This is to study the structure of each proteins, where they are formed, whether they are organellular protein or nuclear protein, whether they are bound to the new membrane or whether they are free in the cytoplasm. So like this, we will consider the overall structurality of the proteins, whether they are glycosylated or phosphorylated. So ultimately, we try to bring the structurality into the proteins, and we will study the structure of the proteins. That is called as structural proteomics, and compare these structural proteomics with other proteins later on. Next is functional proteins. Once we know the structure of each protein specifically, then we will try to find out what is the exact function of this proteins. So this includes the characterization of the enzymes as well as its interaction, post translational modifications, and its effects. Is that clear? So all these analysis is studied under one umbrella, that is the proteomics. Okay, so three types of proteomics. Remember, one is to study the protein expression proteomics, structural proteomics, and functional proteomics. So where do we use this proteomics methodology, or proteomics is applied? So first is protein expression profiling. This can be used in medical microbiology. Okay, so to see how exactly the different bacteria express their protein. Next is when they are expressed, what signals they produce and what disease they can be resultant for. So this is protein expression profiling. Next is post-translational modification. So if we want to study that, we have to study whether it is, in what way it is glycosylation or phosphorylated or proteolylated or digested. All this comes under protein translation modification. Next is proteins interaction. So we study through yeast to hybrid system, co-precipitation methodology, and phage display, especially in case of viruses. Then structural proteins. In this we will study whether the, as I discussed, organellar composition, subproteome isolation, protein complexes. In functional, we use the yeast genomics analysis, affinity purified protein complexes, mouse knockouts, stopping the expression of a gene, and study how exactly that protein will be responsible for a disease or normalcy. Then proteome mining for drug discovery, target identification, validation, different display. So out of this, at least try to divide this, put it into a format, and try to write at least 5 to 6 applications. So this is about the proteomics. So the question will be, discuss about proteomics and its applications. Okay, so you have to write what is a proteomic, what is a proteome, and what are the different aspects of proteome, and what are the different types of proteomics and its application. Is that clear? Okay, so remember, out of the 3 omics, you will be having 2 questions. One will be for 3 marks, the other one will be for 5 marks. So will be there, you just have to note it. Okay, thank you all. Just go through the video. Thank you.